like to begin with a few announcements to make for the class. The announcements will be posted usually to my right on the board. The first announcement is that we have an individual who is a retired community college instructor. His name is Mr. Terry Kern, and for many years he has run a review session for the plant section of Bio 1B, and students have found it very useful. He has sample questions. You get a different sort of take on the material, and students have replied when we've gotten reviews that it's something that really made the class very helpful and very useful for them. So I want to encourage you, if you can make it, his office is upstairs where we have our regular office hours in 5045, and he'll be there from 10 to 11, plus maybe he'll stay longer, and he will start today. So if any of you want to go up there after lecture just to see what it's like, it's certainly worth a visit. The second point to make is that I want to remind you to read your lab for next week before you arrive at the lab. And one of the things you will find in this first lab is that you're being asked to collect a bryophyte. Many of you will never have seen a bryophyte. We will have no idea where the bryophyte is growing. And you'll probably, in many cases, collect something that's not a bryophyte. Don't worry. The whole point of this exercise is to get you to begin thinking about where a bryophyte might grow, what it might look like, and bring it into the lab. No one's going to laugh at you if you bring something in that's not a bryophyte. If it's a snail or a frog, maybe you'll be downgraded. But other than that, try to think about where these organisms might be growing. Okay, today what I'd like to do is to continue talking about the fungi, which I introduced you to on Wednesday. We said that the fungal body was composed of filaments. And biologists give a special name to the filaments that compose the fungal body. We call these filaments, a single filament is called a hypha. If you have several filaments, together, we add an E onto this and we call it hyphae. So a number of filaments together we call hyphae. And if you have many, many hyphae, then we call this a mycelium. Now, if we look closely under a microscope for an individual filament, as I drew it last time, we said it was made up of a number of cells. And if you look inside each of the cells with a microscope, you'll find a single nucleus. This nucleus is haploid, which is abbreviated as N. For those of you who are a little rusty on haploid and N, you might want to look at earlier chapters in the book, which re-familiarize you with this term. Because the nuclei is one nucleus per cell, we give this a name, and you'll see why we're going to give it a name in a moment. We call this particular hyphae, we say that it is mono, because of the single nucleus, karyotic. Monokaryotic. Karyotic is a term which is from the Greek, means nut, but in this case it applies to the nucleus. Or we can call this particular stage the monokaryon. Now, reproduction in the fungi, as I mentioned last time, can take two different forms. It can be either sexual or asexual. In the asexual stage, which I'd like to discuss very briefly first, we can have a new organism being produced by a fragmentation of the monokaryotic hyphae. In other words, a piece of it breaks off and a new organism forms. The other way we can get asexual reproduction is through the formation of spores, which we mentioned last time. And you're going to see spore production in the lab, and I'm going to show you some pictures of spore production in a moment. Now, in this monokaryotic state, the fungus can continue to exist in the soil for many, many years. So in the monokaryotic state, which I'll just abbreviate as MK, And while it's in the soil growing, it is secreting enzymes into the soil, digesting carbon compounds, and then reabsorbing them back into the fungus. We said that the sexual life cycle exists in the fungi, but it's a little different, and in fact very different from many other organisms, because in the fungi there are not two sexes as there are in humans, but there are many sexes. And we call these sexes mating types. Call these sexes mating types. And in the fungi, the mating types can be many depending upon the species. And what we can say happen is that the sexual part of the life cycle begins when two compatible mating types meet and fuse. And let's talk about what happens in this particular situation. So we'll draw two different hyphae, and again, each of them are divided into cells, and into each cell we'll put a nucleus, as we did before. And for our discussion, we will just indicate that these two mating types are plus, and this one is minus mating types. Now, into this particular situation, once they meet and they fuse, what we have occurring is a new hyphal strand, which again has cells in it. But now, if we look carefully at the condition of the nuclei in the cells, we find that the nuclei do not fuse. Even though the mycelium, the two hyphae, have fused, the nuclei have not fused. And we call this particular condition a dikaryon, just like the monokaryon. A dikaryon, two nuclei. And it is a unique, although I've been told a few other organisms produce this, but for our purposes, it is a unique process which the fungi have, that is retaining two separate nuclei of the two sexes without joining them together. And we indicate this dikaryotic condition as N plus N. The fungus can exist for many years as in the monokaryotic state. It can exist for many years in this dikaryotic condition, exploring the soil, secreting enzymes, digesting the carbon compounds in the soil. But then, following the receipt of some signal, and this signal could be maybe running out of food in the soil, or maybe the moisture level of the soil is being produced, we now begin to complete the sexual life cycle. So, what we're going to say here is now, following a signal, the dikaryotic hyphae change their development. And for the purposes of today's discussion, they make a structure, which all of you are familiar with, I think. They make a structure which biologists call, it's kind of a funny name, they call it a fruiting body. And all of you have seen these fruiting bodies. When you go to the store and you buy a mushroom, this is a fruiting body. And if you take this mushroom and cut it in half, and you could put the mushroom under a microscope, you would find that it is made up of dikaryotic, N plus N hyphae. Every place in this mushroom except one place is N plus N. And that one place that is not N plus N is on the structures which are known as the gills, the very fine little tissues that line the underside of the mushroom. On the gills, we have the completion of the sexual life cycle. Because on the gills, the two N plus N nuclei fuse to make the 2N structure. This 2N structure occurs underneath. It exists for a very short time, but during the time that it exists, we have the opportunity for sexual recombination and for the production of variation in future generations. Following the production of the 2N individual or 2N structure, we get meiosis. Again, if you're unfamiliar with this term, go back to your textbook or your high school textbook to produce N spores. And these spores are plus and minus, just as we had in the beginning. And then these make new hyphae. Now, fungal spores are tiny, very, very tiny. And the way most of them are distributed is by the wind. They're carried away by the wind and distributed over vast areas. And billions of spores are made by the fungi. But many of the fungi have also developed rather unusual mechanisms for the dispersal of their spores. And I'd like to tell you one of these mechanisms. It is found in a fungus, which some of you may have heard of before, and for which there is a YouTube link on your assignment sheet to let you see this fungus in action. This fungus is known as Pilobolus. It is a small fungus, maybe 5 to 10 millimeters in size. Now, a couple of things you need to know with regard to this fungus in order to appreciate the way its spores are distributed. First thing you need to appreciate is that this fungus grows on dung, that is, the excrement of animals. If we look carefully at this fungus, we find, again, it's about 5 to 10 millimeters in height. It's divided into 3 general regions: a stalk, a swollen region, and a spore mass. Now, the other fact, aside from that it grows on dung, that you have to know in order to appreciate how the spores are distributed in this fungus, is that this fungus bends toward the light. In other words, it shows phototropism, which we're going to talk about later quite a lot. The fungus shows phototropism. So let's now discuss how this life cycle depends on the distribution of the spores. So the very first step is that the fungus bends toward the light. So here we have the dung, and what you find is that the fungus bends toward the light. It's the very first thing that you see happening. Secondly, the swollen region increases in pressure, and this results in a discharge toward the light of the spore mass, which may land on a plant. The next step is that a cow or some other animal comes walking by and eats the plant. The cow goes about its business, traveling around to lots of different areas, wherever it happens to be, and in the process of moving about, has now changed the location of the original spore mass from where it was produced to someplace else. The cow produces excrement, the spore mass comes out, and we now have a elimination of the spore mass. And we begin the life cycle again. The Pilobolus germinates in the dung and repeats the entire process. My question to you is, for this life cycle to be effective, and this is the kind of question you would have on the exam, for this life cycle to be effective, why does the Pilobolus have to show phototropism? Why is phototropism necessary? Think about this. So, this is both the sexual and the asexual life cycle of the fungi. They are extremely interesting organisms, and they are important, as we said, in many aspects of our lives, and increasingly, as more discovered, there'll be many more properties which come from these organisms. Now what I'd like to do before I show you the slides, I'm just going to put the screen down, is to talk to you about an association of the fungi with another group of organisms. Fungi associate with many different groups of organisms to form unique relationships. And one of the most unique associations which occurs is between a fungus and a photosynthetic organism, either a green alga, which we're going to talk more about in a moment, or a cyanobacterium, or maybe with several photosynthetic organisms. This association produces an organism which you know of as a lichen. A lichen. A lichen is a symbiotic relationship between a fungus and a photosynthetic organism. The green alga or the cyanobacterium provide fixed carbon. And the fungus provides a structure, like a housing, and it absorbs minerals. Lichens are very common. They're very slow-growing organisms, but they have great importance for us because in areas which suffer massive disturbance, for example, for those of you who come from the Pacific Northwest, when Mount St. Helens erupted, the lava completely obliterated all life, covered all soil. What was found is that among the first organisms to come into this area were lichens. They established themselves on the lava, and importantly, through the production of acids, they began to convert the lava into soil, allowing then for later and larger plants to come in. Secondly, lichens are very sensitive indicators of atmospheric pollution. If you go someplace and you don't see any lichens growing, you can probably fairly safely conclude that there is something in the atmosphere which is considered toxic for these organisms and potentially toxic for you. So if you see a lot of lichens growing, that's a great place to live. All right, let's look now at some slides of the fungi and see if I can put the lights down here. I don't know if this does anything. Does that do something? All right, here we go. So here we see a fungus which has the monokaryotic hyphae, and at the end you can see these dark structures. These dark structures are the masses of spores which are at the end of the monokaryotic hyphae, and each of the spores which is being produced is haploid. And if we look at the end of it, we can find this large structure which we're going to look at and name in the lab, and you can see the many spores which are haploid and distributed by the wind. Here we have now the fruiting body, or the toadstool, as some of you will notice. This is all dikaryotic hyphae except for under the gills. And when the spores are produced, they are released and carried away by the wind. Notice if you harvest the fungus that you're really leaving most of the body in the ground. Most of the mycelium is in the ground. Now, if we make a parallel cut to the surface of this particular fruiting body, and we look at that parallel cut, we have the central stalk, we've got the edge of the cap, and now you can see the gills, those very fine lines that are going from the perimeter to the center. And if we look at the gills in high power, you can see the spores that are being produced. So all of this structure here is dikaryotic. The N plus N have already united here. Meiosis has occurred to produce now these N spores, which you can see in this particular slide over here. This would have been the structure that originally was 2N and then underwent meiosis to make the N spores, the plus or the minus. The life cycle, again, is not very complex, involving the two different mating types, the fusion to form a dikaryotic hyphae, and then the production of spores via meiosis. And you're going to study this in the lab. Here is the pilobolus, the structure which is phototropic. Here is the dung, and the light is coming from this direction over here. The spores are discharged as a result of the swollen mass. And by the way, this swollen mass is very, very unique because it focuses the light. It acts as a lens. And to remind you, this web address for looking at the shooting off of the spore mass is given to you in your notes, and it's not 10 or 15 seconds long. Have a look at it. And finally, the last slide I want to show you is of a lichen. Here is a slide of a picture that you probably have, many have seen your own way, personal experience, where we have this organism which consists of a fungus, or maybe several different species of fungi, plus one or more species of algae or cyanobacteria, photosynthetic organisms, growing on this rock, secreting acid, and eventually decaying the rock, causing it to produce what we would consider to be soil. Okay, let's try this over here now. Screen up. Give that a try. I don't know if it's going to come up here. Oh, yes? I don't seem to be able to get the lights. Can you do the lights? There we go. I think the chalk prevents you from getting lights. Okay, so these are the fungi, and they are a very unique group of organisms for which there are entire classes here on the campus, if any of you are interested in pursuing these studies further. They're really fascinating and not very well studied, considering how many of them are there. Now what I want to do is to move on to the next group of organisms, which in this case represents the first true plant that we're going to be dealing with. The fungi, I said, were studied with plants for historical reasons, but are not plants. They're not related to plants. Today what we're going to do is consider the first plants. Now, the first group that we're going to consider today, which is known as the algae, represent the first plants, and like the fungi, many organisms were grouped by Linnaeus into the algae because to him they appeared to share a number of common characteristics. But again, based upon more recent studies, recent forms of looking at things, including molecular forms, many of the organisms which were originally grouped with the algae have now been moved different places on the eukarya branch. But for our discussion today, we're going to again consider them as one unified group in order for us to be able to at least make some generalities. There are about 130,000 species of algae, at least that have been identified, probably more that remain unidentified. Linnaeus classified them based upon some characteristics that he thought would make them part of this group. Many of the other ways of classifying the algae depend upon other characteristics. For example, color. If you look at the algae, you find that they can fall into 4 different groups based on color. Brown algae, red algae, the green algae, and a group which we'll call yellow, which consists of varied species. And we'll talk about this in a moment. So another way of classifying the algae is based upon color. Another way of looking at whether they belong together or in the same group is to ask whether the algae have any way of moving. So we might talk about movement. Some people say, do they move or do they not move? We'll divide the algae based upon this. Movement implies some kind of locomotion. And in order to get locomotion in the algae, we would have a structure which moves. And many of you have heard of this structure before. It's called a flagellum, or plural flagella. Does the organism have flagella? That's one way of looking at classification. But for our discussion today, and to make things a little easier to handle with this 130,000 species, I want to make another way of dividing the algae into two separate groups. The first group that we're going to divide them into is that organisms which are primarily or exclusively single cells. Versus organisms that are usually multicellular. Into these sort of artificial divisions, we're going to put two groups, and we're going to talk about these groups in a moment. We're going to put the diatoms, which are exclusively unicellular, and a group which probably most of you haven't heard about, known as the dinoflagellates. Into the group which is primarily, if not exclusively, usually multicellular, we're going to put the green, the red, and the brown algae. Now let's talk about the first group, which consists of organisms that are primarily, if not exclusively, unicellular. These organisms, for our point of discussion, consist of the dinoflagellates and the diatoms. If you add a third group to this, that is the small marine invertebrates, together, if we put all these together, these make up the plankton in the ocean. They are the base of the food chain. And as such, they are extremely important because all of the other organisms in the ocean depend upon whether or not these guys exist. Very important point to make. The second important point to make about this group of organisms, and in particular about the diatoms, is that it has been estimated that 50%, some people say 80%, but 50% of the oxygen renewed in the atmosphere comes from the activity of the diatoms. So these are extremely important organisms based on food and based upon the regeneration of the atmosphere. And hence, the health of the oceans is really important for life to continue on Earth. It's not just what goes on land. Now what I'd like to do is to talk about these two groups a little more closely, and let's look at the dinoflagellates. The dinoflagellates tend to grow along the coasts in shallow, sort of gently flowing or still water. And they are more or less in harmony with their environment. They don't make too much, they don't make too little. But at times, when there is a lot of rain on the land and a lot of nutrients are washed into the water along the water's edges, the dinoflagellates can grow tremendously because they now have a big supply of food. And when this happens, when they grow tremendously, we call this growth a bloom, an algal bloom. You know of this algal bloom in a more common parlance, this algal bloom is a red tide. Now, why do we as humans take so much interest in red tides? It is because the dinoflagellates produce a nerve toxin. And when we have the algal blooms, when there are many, many of these organisms, what happens is that the fish ingest very large amounts of the dinoflagellates and then die. And you have massive fish kills. But you'll also notice, for those of you who've had experience with red tides, that they close the beaches when we have a red tide. And the reason they close the beaches is because not only do the fish eat the dinoflagellates, but the shellfish eat the dinoflagellates. And the shellfish have a particular response. They are not as sensitive to the nerve toxin, maybe not be sensitive at all. And so they can accumulate large amounts of the dinoflagellates and still live. And when humans come along and see what looks to be a healthy shellfish and harvest it, they then, the human then gets a huge amount of nerve toxin and can die from it. So beaches are closed because of the dinoflagellates, which can have great economic impact by killing the fish. Now, the second group of organisms which make up the unicellulars are the diatoms. I think if I were to begin biology again, I would spend more time studying these very unusual organisms. The reason why they are so unusual and so beautiful is that they have impregnated into their cell walls silica, which gives them remarkable, almost snowflake-like, and I'll show you some pictures, designs. This makes the walls of the diatoms very hard, very, very dense. And as a result, when the diatoms die and when they settle to the base of the ocean, over millions of years, they can accumulate, these are the fragments of the walls, to very, very great depths, great amounts. And here's an example of material which represents the accumulation of diatoms. This is known as diatomaceous earth. And diatomaceous earth has many, many practical uses, many applied uses. You use it daily if you brush your teeth. The small abrasive, which is in your toothpaste, represents the walls of diatoms. If you ever have silver, which you may not have, and you polish it, the small abrasion is a result of diatoms. Filtration systems that are very widely used are made of diatomaceous earth because the small pores between the cell walls allow for marked and great filtration. So these organisms are of extreme importance. They are ones which we daily use but not realize where they come from. Now what I'd like to do is to consider the organisms that are usually, if not exclusively, multicellular. And as we said, these organisms are the red, the green, and the brown algae. The red algae predominate in the tropics, in warm water. The brown are the algae which form the kelps that are off the coast of Northern California, and they tend to grow in cold water from about mid-California up through Alaska, the kelps. And then we have the greens, which we're going to talk about in a moment. The algae are commercially very important. If any of you are from Asian cultures, you probably have used algae as a source of food. It contains a rich supply of trace elements and minerals. Algae also has a large use with regard to products that are produced from the algae and, again, used in many of our daily applications. A compound or group of compounds known as alginates are extracted from the algae, and these are used for food thickening, food thickeners, or emulsifiers. And for those of you who have ever worked in a lab, you have daily probably encountered a product of the algae, namely agar. Agar is or originates from the algae. Now, not only are the algae multicellular, but in this particular kelp that we have here, they are now differentiated. They are now forming different cell types in the multicellular form. And this particular alga, which you can find growing off the coast, certainly in Monterey and perhaps up here as well, looks like a small palm tree. And I want to just point out to you a couple of regions on this particular alga. It has a central structure here. You don't have to write down the names, which is called a stipe. And then when it's growing in the water, it has a way of anchoring itself to the rock. So it has this structure which looks like a root. We call it a holdfast. And this allows the alga to stand up in the tide. And then it has these structures at the top, the blades, which actually carry out the photosynthesis. So in the multicellular algae, we not only have a lot of cells making up the organism, but we also have the cells differentiated into another cell type. In this particular kelp which we have here, if you come up and look, you'll notice in addition to the blades that beneath or associated with the blades are large structures which look like balloons. These are bladders and allow the alga to remain rooted, so to speak, at the bottom, but to float up to the surface of the water to be able to photosynthesize. So these are important organisms with regard to their ability to take advantage of their environment and also be able to take the sunlight by floating up near the surface. The algae are an important group of organisms, and they are important because not only economically, but for our purposes, they're important because they have originated the life cycle that we are going to see exists for the rest of the time in this class. The life cycle that all the plants outside have can be traced back to the green algae. And what I want to do now is to introduce you to this remarkable life cycle, this life cycle that most of you didn't even suspect existed. And in fact, if you were writing a science fiction movie, you couldn't dream up of a life cycle more unique and more weird than what I'm going to tell you about now. How's that for an introduction? Okay. Let's begin, in order to appreciate this life cycle, let's begin with our life cycle. So our life cycle consists of two sexes, a male and a female. And each of these individuals is diploid, 2N. That is diploid. Again, if you're unfamiliar with this term, look it up in the textbook so that you're familiar with it. Through the process of meiosis, we produce the gametes. And those gametes, in the terms of the male, are the sperm, which is now N, because meiosis has occurred. And for the female, the egg, which is also N. And then what happens is that we have fertilization. And you make the new 2N individual. The new 2N individual. Repeats the cycle again. Now, what have the algae done? This particular life cycle that I'm going to show you originated several times in the green algae. Not all of the green algae have it, but it originated several times independently during evolution. And we don't know what the forces were, evolutionary forces were at the time that caused this life cycle to originate. But they must have been very strong. They must have favored this life cycle in order for it to have originated independently several different times. So let's begin this life cycle exactly the same place. And we're going to start with one individual in this case. Let's start with a 2N plant. For this 2N plant. Now, this 2N plant, just like you saw with us, is going to have meiosis occurring. And we're going to make an N structure, just like you saw with the gamete. But this N structure is not a gamete. not a gamete. In fact, what happens is this end structure develops into another organism. Another organism. A whole new organism develops from this. This is a spore. And this spore develops into another organism. Because the spore is N, this new organism is also N. So you have an N individual produced. Now, what happens is, and let's just say for the sake of our discussion, that this other organism now produces sperm and egg. We can have sperm being produced by one organism and egg by another organism, but now for our discussion, let's just say they're produced both by the same new individual. This is N. The sperm and the egg are both N, so all we have to have occurring here is mitosis. No meiosis is necessary. In fact, we can't have meiosis. Then at this point, we have fertilization, which makes the new 2N individual. So what the algae have done, the green algae, have developed a life cycle in which fertilization is delayed. It's delayed. And it is delayed by the production of a new organism. Very important, very unique life cycle. Why would you want to delay fertilization? Aren't you putting your next generation at risk by delaying fertilization? We can talk about this more. We have names for these two generations. The one which produces the spore, we call this plant the sporophyte. That is, it makes the spore. Makes the spore. We call it the sporophyte, and it is 2N. The one which makes the gametes, this other organism, makes the gametes, we call this one, I'll put it up here so it's not, we call this other one, we call the second plant, which is haploid, the gametophyte. So we have an alternation of two different generations of plants for the life cycle to be completed. And as a consequence, the life cycle is known as the alternation of generations life cycle. Thank you. Thank you. Okay, so the point I want to make is that these organisms live in the ocean. And because they live in the ocean, they are surrounded by water. And the gametes are able to get together by swimming. They have flagella. And this is important because later on, when we move onto the land, and you look at the land plants, they don't have water surrounding them. So how do the gametes get together? So I want you to keep that in mind. Let's look now at some slides of these various items of the algae. And I'd like to begin by showing you a slide of one of the unicellular or single-celled algae. These would be ones which have flagella on them. They are totally contained unit. Given nutrients, sunlight, and water, they're perfectly happy. This particular alga is a single cell, looks like two airplanes crashing. The nucleus is in the middle here. This is a unicellular green alga. Then the algae can begin to form colonial forms, and you're going to see some of these in the lab, where we have several cells getting together on the way to multicellularity until we can form quite large, complex colonies of the green algae. And again, you're going to see some of these in the lab, upwards of 60,000 cells perhaps. The colonial algae can also take the form of a filament, and you're going to look at this filament in the lab where individual cells are joined together to make a multicellular organism. Here is the plant that I just showed you, the brown alga that I just showed you up in front, and we have it here. This is this root-like structure which holds it to the rock by the tide pools, a stalk which is given the name stipe, and then the region at the top where photosynthesis occurs. Now let me show you some of the diatoms. As I said, if I were to study again, I would spend more time learning about diatoms. This beautiful structure, which is very unique, is all dependent upon the deposition of silica into the cell wall. And when the organism dies, these individual cell walls settle to the bottom of the ocean and there accumulate to great depths. Now, in Victorian times, before there was the web and television and even radio, people used to have other types of entertainment. And one of the types of entertainment was to arrange diatoms on slides into various patterns. And it's very relaxing, guys. You might think about it. Okay. Here's another one. And they would have contests, too. The Victorians were very unusual people. They would have contests, too. Here's another one. Very beautiful. And a lot of people made their family crest this way, too. Here is a picture of the dinoflagellates. These are the organisms which cause the red tide. And again, they have flagella, which is how the word flagellate appears in their name. And as a result of their massive accumulations, this large amount of nerve toxin is produced, and people die. And finally, I want to just show you an application, a practical application of the algae. This is a picture taken from the air in Australia. And to give you an idea of the size, here is a house over here next to this arrow. These are ponds of algae. And as the ponds dry out, or as the salt is increased in the ponds, you can change the color. That is, the pigments which are present in the algae. And these pigments then are used in many different forms of cosmetics, in our drugs, in the vitamins, in the pills you take. And they are all natural products, and they come from the algae, and so they are easily used without any controversy. Okay, visit Mr. Curran if you want, and I'll see you all on Monday.